Hi everyone, welcome back to the Ventistega. If you're new here, this channel explores the fascinating evidence for evolution through genetics, paleontology, and comparative biology. In previous videos, we've looked at chromosomal evidence like the human chromosome 2 fusion. Today, we're exploring something even more abundant in our genome, pseudogenes. So what exactly are pseudogenes? Simply put, they're broken genes, sequences in our DNA that look like functional genes but can no longer produce working proteins. They're like genetic fossils that have accumulated mutations over millions of years, rendering them non-functional. But here's the crucial question. Why would these broken genes exist in our genome if we were independently designed? And more importantly, why do we share the same broken genes with other primates at the same locations in our genomes? Let's start with the basics. To understand pseudogenes, we first need to understand how normal genes work. A functional gene contains a promoter region that signals where to start reading, coding sequences, called exons, that contain instructions for making proteins, and a termination signal that tells the cell when to stop. When a gene is expressed, it's transcribed into RNA, then translated into a protein that performs some function in your body. Now, here's where pseudogenes come in. Pseudogenes are DNA sequences that resemble functional genes but have accumulated disabling mutations. These mutations can include premature stop codons, like hitting a period in the middle of a sentence, causing protein production to halt too early. Frame shift mutations, insertions or deletions that throw off the entire reading frame, scrambling the genetic message. Deletions of critical regulatory regions, removing the on switch so the gene can never be activated. Or broken splice sites, disrupting how the gene is processed into a final message. The human genome contains approximately 20,000 functional genes, but over 20,000 pseudogenes. That's roughly one broken gene for every working one. Now, let me give you one of the most powerful examples, the vitamin C pseudogene, also called GULO, L-gulono gamma-galactone oxidase. Here's the story. Most mammals can synthesize their own vitamin C. They don't need to get it from their diet because they have a functional GULO gene that produces the enzyme needed to make vitamin C. But humans can't do this. We need to eat foods rich in vitamin C, or we develop scurvy, a disease that plagued sailors for centuries. Why? Because our GULO gene is broken. It's a pseudogene. But here's the evolutionary prediction. If humans share a common ancestor with other primates, we should find the same GULO pseudogene in related primates, the same disabling mutations in the same locations, and additional unique mutations in each species that accumulated after they diverged from common ancestors, like branches on a tree accumulating their own specific damage over time. And that's exactly what we find. Humans, chimpanzees, gorillas, orangutans, and other primates all have a broken GULO gene. But it's not just broken, it's broken in the same ways. We all share the same critical deletion in exon 10. We all have the same frame shift mutation. The pseudogene is located at the same chromosomal position in all these species. But each species has also accumulated its own unique additional mutations since diverging from common ancestors, like branches on a tree accumulating their own specific damage over time. The probability of this pattern occurring by chance or independent creation is astronomically small. It would be like finding the same typos in the same locations in translations of an ancient text. clear evidence they were copied from a common source, not independently created. Let's look at another powerful example: olfactory receptor genes, the genes responsible for our sense of smell. Humans have approximately 400 olfactory receptor genes, but here's the shocking part. About 60% of them are pseudogenes. That's over 400 broken smell receptor genes. Compare this to dogs, which have a much smaller percentage of olfactory pseudogenes, or to mice, which have even more functional smell receptors than dogs. Why do humans have so many broken smell genes? The evolutionary explanation is straightforward. Our primate ancestors transitioned from relying heavily on smell to relying more on vision, particularly color vision. As our ancestors evolved trichromatic vision, the ability to see red, green, and blue, the selective pressure to maintain every single smell receptor decreased. Without selective pressure, mutations that broke these genes weren't eliminated by natural selection. Over millions of years, these genes accumulated disabling mutations and became pseudogenes. This is a classic example of use it or lose it in molecular evolution. The pattern we observe: primates that rely more on vision, like humans and apes, have more olfactory pseudogenes. Primates with better color vision have more olfactory pseudogenes. Animals that rely heavily on smell, like dogs, have fewer olfactory pseudogenes. This nested hierarchy perfectly matches evolutionary predictions based on our understanding of primate evolution. So why are pseudogenes such powerful evidence for evolution? 1. Common descent prediction. Evolution predicts that species sharing a recent common ancestor should share the same pseudogenes with the same disabling mutations. This is exactly what we observe. If species were independently created, there's no reason they should share the same broken genes in the same locations with the same mutations. 2. Molecular clock. Pseudogenes also serve as a molecular clock. Because they're non-functional, they accumulate mutations at a relatively constant rate. They're not subject to selective pressure. By comparing the number of unique mutations in a pseudogene across species, we can estimate when those species diverged from their common ancestor. These estimates align beautifully with fossil evidence and other genetic data. 3. Nested hierarchy. The pattern of shared pseudogenes creates a nested hierarchy that perfectly matches the evolutionary tree constructed from fossil evidence, anatomical comparisons, other genetic data, and geographic distribution. All independent lines of evidence converge on the same tree of life. 4. Functional genes in other species. Many of our pseudogenes are fully functional genes in other species. For example, our broken GULO gene is functional in most mammals. Our broken olfactory receptors are functional in species that rely heavily on smell. Our egg yolk protein pseudogenes are functional in egg-laying animals. This pattern only makes sense in light of common ancestry and evolutionary change. Now, let me address some common objections. Objection 1. Maybe pseudogenes have hidden functions we haven't discovered yet. While it's true that some pseudogenes or their RNA transcripts have been co-opted for regulatory roles, the vast majority are demonstrably non-functional. We can show this because they accumulate mutations at the neutral rate, no selective pressure. They have disabling mutations that would prevent protein production. They can be deleted in organisms with no observable effect. And the pattern of shared disabling mutations across species makes no sense if they were functional. Even if some have been co-opted for new functions, this doesn't explain why we share the same broken genes with the same mutations in the same locations as other primates. Objection 2. Shared pseudogenes could be the result of a common designer using similar blueprints. This explanation fails on multiple levels. Why would a designer give us broken genes at all? Why would a designer give us the same broken vitamin C gene as chimps, making us both dependent on dietary vitamin C? Why did the patterns of mutations match the prediction of common descent and not common design? Why would a designer create hundreds of broken smell receptor genes in humans, but working ones in dogs? The common designer explanation requires us to believe the designer intentionally created non-functional genes with a pattern that perfectly mimics evolutionary descent. This is indistinguishable from common descent and requires additional, unfalsifiable assumptions. Objection 3. The genome is too complex. We don't understand everything yet. This is an appeal to ignorance. While it's true we're still learning about the genome, we understand enough to recognize that pseudogenes are indeed broken genes. The evidence is overwhelming. Experimental verification shows they don't produce functional proteins. Molecular and biochemical analysis confirms the disabling mutations. Comparative genomics shows the pattern matches evolutionary predictions. Population genetics shows they evolve neutrally. Saying we might discover something later doesn't negate the mountains of positive evidence we have now. So there you have it. Pseudogenes are molecular fossils embedded in our DNA, providing powerful evidence for human evolution. Just as paleontologists study fossilized bones to understand evolutionary history, geneticists study these molecular fossils to trace the history of life at the DNA level. The pattern is undeniable. We share thousands of pseudogenes with other primates. We share the same disabling mutations at the same locations. Each species has accumulated its own unique mutations since divergence. The pattern creates a nested hierarchy that perfectly matches the evolutionary tree. Pseudogenes aren't just evidence for evolution, they're evidence that demands an evolutionary explanation. Any alternative hypothesis must explain why we share the same broken genes with the same mutations in the same locations in a pattern that perfectly mirrors evolutionary predictions. If you found this video informative, please give it a thumbs up and subscribe to the Ventastica for more content on the evidence for evolution. In the comments, let me know, were you aware that we have so many broken genes in our genome? What other genetic evidence for evolution would you like me to cover? Next time, we'll explore another fascinating genetic evidence: endogenous retroviruses, viral DNA that's been permanently inserted into our genome and shared with other primates in the exact same locations. Thanks for watching, and I'll see you in the next video.